Hello, in this video I will present you the web application PAVOOC and explain how you can use it. Using PAVOOC, which stands for Prediction and Visualization of On and Off Targets for CRISPR, you can design domain-aware CRISPR sgRNAs for gene knockout and gene editing experiments. Right now you can see the start screen. Let's first design a gene knockout experiment, so we can leave this radio box as is. Right below, you enter the genes which you want to knock out. Use either gene symbols or ensemble IDs. You can also paste a comma or space-separated list to add several genes at once. If you are working with cancer cells, you can select your cancer cell line in the input field on the top. The application then filters mutation-affected single-guide RNAs and also notifies you about copy number alterated regions which might affect your experiment. In the next screen, we see a pre-selected list of sgRNAs for each of the genes. These recommended sgRNAs are selected based on a combination of cutting-edge on-target efficacy prediction scores and the off-target CFD score. This helps minimizing off-target and maximizing on-target effects. You can also see that KRAS and CDK4 are both in a copy number alterated region in the selected cell line. It is now possible to view and edit the recommendations. After the page is loaded, we see three main views that all serve to display information about the available sgRNAs. Have a look at the protein structure first. The selected 2W96 structure shows CDK4 in complex with a D-type cyclin. As we are interested in CDK4 only, the other chain is grayed out. Some of the amino acids in the bluish CDK4 chain are highlighted. These correspond to the cut positions of available single-guide RNAs. On the right, we can see a list of these, with information like containing domain, on-target and off-target score. Taking the sgRNA at the top as an example, we can see that it has a very high on-target but a rather low off-target score. The scores go from 0, which is bad, to 1, which is good. This sgRNA is very likely to cut the targeted site at a very high rate, while it may also cut at some off-target sites due to the rather bad off-target score. At the bottom, you see a genome sequence viewer with additional information like exons, domain regions, available PDB structure files, and copy number amplification. All three views are connected, so hovering an sgRNA in one view highlights it in the other ones. While not hovering anything, the selected sgRNAs are highlighted. The shown PDB structure can be changed with a click on the PDB name in the title or in the sequence viewer. The label of the CNS track at the bottom indicates the severity of the copy number alteration for the currently selected cancer cell line. To easily evaluate the effect of different cancer cell lines to the edited gene, you can change the cancer cell line at the top. CDK4 seems to be a gene which is affected by copy number alterations in various cancer cell lines. To improve the pre-selected guides, you can use the checkboxes in the list on the right. Use the headers at the top of the table to add filters or change the sorting columns. CDK4 contains one domain only, the P kinase domain. By using the filter and sort function, it is possible to target specific domains for genes that contain several ones. It is also possible to change the proportion at which the different scores contribute to the sorting. Harnessing all the combined information, you can now select the optimal sgRNAs. Back in the overview window, you can see and control your adjustments. Use the detail view to gain trust in the computed recommendations or to employ your own sgRNA selection strategy. Then download the library as a CSV file to perform your CRISPR experiment. If you have any questions or comments, contact me as pointed out in the video description. Thanks for watching and have fun!